Hello guys, it's Mr. Y. Today we're going to be talking about Punnett squares as part of our continuation of the genetics unit. The learning objectives for today, you're going to learn how chromosomes are related to genes and also how genes are related to this term called alleles. Terms like dominant and recessive, which hopefully you have already learned about in middle school, but if not, you'll learn about that here. How that connects to things like Punnett squares, and how probability is correlated to Punnett squares, and also terms like genotype and phenotype and homozygous and heterozygous, if you've never heard of those terms before either. Okay, so without going into too much detail here, every species has a set number of chromosomes for the majority of the cells in its body. As I said, most species can be thought of as having a set number of chromosomes. So for instance, for dogs or wild dogs, like coyotes or hyenas, wolves, they typically have somewhere around 70 to 80 chromosomes per body cell. Cats, on the other hand, have usually somewhere in the range of 30 to 40. And by cats, I mean anything from lions to your typical house cat. Even yeast cells that we use to make bread have 32 chromosomes. Carp, this is a type of fish, has 144 chromosomes in its body cells. And algae, pond scum, has 148 chromosomes in its typical body cell, I guess if you want to call it that way. And so what I'm trying to point out here is that the number of chromosomes does not correlate to how complex an organism appears to be on the outside. For humans, the magic number for us is, of course, 46. Now, we usually talk about this in terms of 23 pairs of what we call homologous chromosomes. But again, the number of chromosomes does not relate to complexity. So something as simple as a pond protus here, this guy sets the record at 15,600 chromosomes in just its one-celled body. So again, number of chromosomes or amount of DNA, if you want to think of it that way, does not correlate to how complex an organism is. When it comes back to humans, though, of course, when we say we have 46 chromosomes, that means that initially, when you were originally that initial zygote, 23 of the chromosomes you inherited from your father's side, the sperm, 23 from your mother's side, the egg. So 23 and 23, of course, gives you 46. And of those 46 chromosomes that are in the vast majority of the cells in your body, 44 of them, in other words, 22 pairs, are what we call autosomal chromosomes. The remaining two, the remaining pair, are what we call the sex chromosomes. These are what determine the genetic identity of offspring. Now, we're just talking genetic identity. We're not talking self-determined sexual identity here of this stuff. But in terms of the genetics, the way it breaks down for humans typically is that if you are female, you've inherited what we call two X chromosomes, XX, one from the mother, one from your father, XX. If you're a male, you are what we refer to as XY. Again, one from the mother, one from the father. But note at this point that what actually ends up determining the genetic gender identity of this setup is actually the father's gametes, because the mother's gametes, the ones from mom, which I've color-coded here, well, the mother's side can only give you an X chromosome or an X chromosome, while the father's side could give you either the X chromosome or the Y chromosome. And again, there's no conscious, choice system here. This is just, all happenstance. But it is the father's gametes, the father's sperm in this case, that determines the genetic gender of the child in that case. So here's a picture of what we call karyotypes. These are all 23 pairs of chromosomes lined up next to each other. So again, they put these into pairs, pair 1, pair 2, pair 3. And you can see them here as well. Again, 22 of these pairs are what we refer to as autosomes. They have lots of genes that code for lots of different proteins that we're still working on discovering. But one pair, you can see right here, looks a little bit different. This is the sex chromosomes. So this is a magnified image of that. You can see there's the X chromosome and there's a Y chromosome. So this individual here would be a male, not a female, genetically speaking. And again, the other ones, they're referred to as autosomes. And you can see that the Y chromosome, while we call it a Y chromosome, really what it is, it's really more like a dwarf X. So you can see the centromere clearly here, and like the centromere might be here in the Y chromosome. It's just that the Y chromosome is missing a lot of DNA. And we'll come back to this when we talk about sex-linked traits. But you can see clearly that if you were to have 2 of these X chromosomes, you would actually have more DNA than having just an X and a Y. So the Y chromosome, while we call it a Y chromosome, really it's more like a dwarf X chromosome in actuality. All right, some terms to clarify. Now, I've been using these terms for a while, but it's nice to get the actual definitions in your notes, so make sure you do that. The term gene. A gene is a segment of DNA that contains coded instructions for a specific protein. And you guys should have, by this point, learned how to do transcription and translation going from DNA to mRNA, mRNA to tRNA, then via ribosomes to amino acid chains and all that stuff. And what we're going to get to now is understanding how these proteins can greatly influence an organism's traits. Whereas a trait is a characteristics. Typically, when we talk about traits at this level, we think of physical traits. That's not always the case, but that's the easiest way to start. So you could think of physical traits like red hair, blue eyes, freckles, things of that nature that an organism can pass on to its offspring. So we don't mean things that can be gained through experience. So for instance, scars, for instance, you don't inherit scars. You gain those over the course of your life. Or language is another one. Like you inherit the ability to learn a language, but you don't inherit a specific language. What language you actually end up speaking depends on where you grow up. So traits can become somewhat of a sticky situation, but if you start thinking of it just in terms of the physical stuff, it makes it a bit easier to understand. Then understanding that there's different forms of the same genes. These are what we call alleles. So different versions of the same gene in the same location that code slightly in different ways and produce slightly different effects. So one of the ones I mentioned earlier was freckles. There is a gene that produces a uneven coloration of the skin, which we call frecklation or freckles, especially on the face. But there's also a gene that helps to kind of limit this and prevent freckles. So there is a gene for what we call freckles, and we're going to use capital F here to symbolize that, and a gene for no freckles, which we're going to use a lowercase f here. And those of you who studied this in middle school know what I'm getting at eventually with this. There's another type of trait that's kind of works this way. All these are greatly simplified in reality, but there are a few that work this way in humans, not many though. This next one is on widow's peak. A widow's peak is a hairline that comes to a point right here. The converse of a widow's peak, the opposite of a widow's peak, would be what they call either a rounded or straight hairline, where it's just nice and even and you don't have that point system right here. So let's assume the gene for widow's peak is going to be symbolized with a capital W, and the gene for no widow's peak, i.e. a straight hairline, would be a lowercase w. And what we see is that sometimes some of these traits show up more often than not. That is to say, some alleles tend to occur and show up and show their effects more frequently than others. The ones that show up most often are usually what we define as being dominant alleles. That is to say, it's an allele whose trait always appears in the organism when the genetic when the gene is present, when the allele is present, I should say. So we typically use capital letters like capital F or capital W. And this is in contrast to a recessive allele. This is an allele whose trait is masked or hidden when the dominant allele is present. I know that seems kind of confusing, but we'll see if we can make it a bit clearer. Typically, though, we represent recessive alleles with lowercase letters, so lowercase f and lowercase w. So let's run through this and make sure it's a bit clearer to you guys. So remember that every organism has at least 2 genes coding for the same protein in most cases. But these genes can be alleles that are different from each other. You can have one allele for one thing and another allele for another thing, even though it's coding for the same basic trait. So for freckles, for instance, a person might be like this. They might have one dominant allele for freckles, and they might have one recessive allele for freckles. And maybe one of these came from the mother side, and one of these came from the father side. It doesn't really matter which one. But they are a mix of these two. And it's understanding terms like dominance and recessive, which gives you hints into how the actual physical appearance of the person will be. So what would this person look like? This person who has one dominant allele for freckles and one recessive allele for no freckles. Their physical makeup, what we call their phenotype, would be to show freckles with this genetic inheritance. Well, let's talk about a different situation. Let's talk about what if this person had in fact had both dominant alleles, both for freckles. Well, what would their phenotype, their physical appearance be? They would obviously show freckles as well. And physically speaking, you would not necessarily notice the difference between these two individuals. You couldn't tell by looking at them which one was 2 capital F's and which one was 1 capital and 1 lowercase. There would be no way to determine that. And let's take on this third situation. What happens if this person had in fact inherited both recessive alleles, both lowercase f's for no freckles? What would their phenotype, their physical appearance be then? Well, they would have no freckles and they wouldn't look like this, obviously. They'd have, nice clear skin as comparison. So what you see by this dominant recessive setup is really there's only 3 possible genotypes for any system when you're using a dominant recessive system. It doesn't always work this way, but when it's really just a pure dominant recessive system, there's only 3 possible outcomes. And I could be using any letter you want. I'm sticking with the capital F's here. It could be any letter you want to choose. The letter itself really doesn't matter. So sticking with letter F, you could have both uppercase, you could have both lowercase, and you could of course have a mix. Now, quick side note, the uppercase always goes before the lowercase. So you'll never see, let me switch to my marker here. You'll never see lowercase, erase that. Sorry, lowercase. You'll never see this. You should never write it like this. We always put the dominant one first. So this does not work. So never see that. So that's why, you don't say, oh, well, there's a 4th way. No, there's not. There's only 3. All right. Go back to my pointer. So there's really only 3 options, both uppercase, both lowercase, and one of each. Again, capital letter gets written first. Now, we've already discussed the possible phenotypes here, and I do recommend you get this table down. The genotype here is the genetic makeup, so both uppercase or both lowercase or a mix. The phenotype is the actual physical traits that the person would express. So the first one, of course, both uppercase, they show freckles. The bottom one, of course, both lowercase, they show no freckles. And when it's a mix, well, this is where you get more clear on the idea of dominance versus recessive. When it's a mix, the dominant one is what gets expressed, hence its dominance. So this person would show freckles, and that would mask or hide the recessive allele right here. It's being masked. That's why it's the lowercase one. And there's 3 other terms you have to get used to with this idea. The terms are homozygous dominant, heterozygous, and homozygous recessive. Now, homozygous means the same, or same zygote, more loosely translated. Same as in both of these are the same case structure. Both are uppercase. And you can see homozygous is used here as well, because both of these are lowercase. And the way you tell the difference between these two is this is homozygous dominant, i.e. both of them are the same and they're both uppercase. Whereas this is homozygous recessive. Both are the same and both are lowercase. So homozygous dominant, the same, both uppercase. Homozygous recessive, the same, both lowercase. The heterozygous one is the mix. Now, we don't say dominant or recessive because you don't really need to. Heterozygous, right here, tells you these are different cases from one another. You have a mix, and it's not, again, said usually, but heterozygous is always going to show the dominant one. So there's no such thing as a heterozygous recessive. It always shows the dominant because the dominant one is, what's going to take over. It's dominant. And there's old terms for these as well. Go back to my pointer here. So homozygous dominant, the original terms from the early 20th century, you might hear as purebred dominant, or for homozygous recessive, you might hear the terms purebred recessive. And let me just write that out. So, like, it's kind of a very artificial term. Can't write with this mouse. So purebred is another way you hear these terms, homozygous, purebred. Whereas Heterozygous, you might hear the term hybrid. And if you think of what a hybrid is, it's just a mix. So heterozygous, you might hear the term hybrid. Purebred is when they say something is homozygous, though purebred doesn't indicate homozygous dominant or homozygous recessive. So just keep that in mind. So let's clarify these other two terms, genotype and phenotype. Genotype, again, is the genetic makeup of an individual, i.e. what letters they actually inherit. And it should be a pairing. So, what do they, what two letters do they inherit? You could have, again, you could have both uppercase, both lowercase, both a mix, a heterozygous. Whereas phenotype is the physical traits that an individual are inheriting. Again, we're not talking about things you gain over the course of your lifetime. We're talking about genetic inheritance. So the easy way to remember this is genotype is genetic, i.e. the letters here. Genotype is genetic. Phenotype is physical. What physical traits are being expressed. That's a very simple way of remembering it. Just keep in mind, phenotype can also be like certain behavior, propensities towards certain behaviors, things like addiction and things like that. So it's not always physically things you can see, but phenotype is what's being expressed physically and in the basic level. Genotype is what's being inherited genetically. So let's run through a few situations where this gets kind of tested a bit. Let's suppose we have two parents. Parent one is homozygous dominant, in other words, two capital F's for freckles. The other parent, parent two, is homozygous recessive. Again, same but lowercase, two lowercase s for no freckles. So one parent has freckles, one parent has no freckles. You might also say one parent is purebred for freckles, the other parent is purebred for no freckles. That would be the old style of saying this. And we want to know what are the odds of any of their kids having freckles. And that's where this little device called a Punnett square comes in. Punnett squares were developed in the early 1900s by a mathematician called R.C. Punnett after trying to work out certain situations with pea plants and with chickens. Punnett was a mathematician. He figured out how to make this out of a matrices. So if you ever go into advanced mathematics, this box system is actually called a matrices, but we call it Punnett squares in genetics. So here's our two parents, capital F, lowercase f. I just brought them down here. We're crossing them. That's what the little x means. And the basic rule is you take one parent and you separate out each copy, each allele, into a different square line. So let me do this as a pen. Capital F here and capital F here. The other parent is going to go along the side. And traditionally they would say the male parent goes on top, the female parent goes on the side. It really doesn't matter, guys. As long as you're separating out the parents and as long as you don't intermix the parents on either side. So one parent on the top, one parent on the side. Don't change that format and you'll be fine. So again, keep one parent at the top, one parent at the side. Now what you have to do is you have to do this crossing effect where you bring down the top and slide over the side. So again, we want to know what are the odds of a kid from these two parents having freckles. We're going to be able to answer this in two different ways. But let's see how this works out. And let me switch to my pen here. Notice I brought the capital F down and the lowercase f over. Capital F goes down, lowercase f again goes over. And then again, you repeat that same thing. The capital F came down. Again, capital letter always goes first. Lowercase f comes over. And then again, one more time, capital F comes down. Just not trying to write over myself. Lowercase f comes over. So this is our results. And let me clear this up now that you've seen this, how this works a bit better. Now, the question, remember, was what are the odds of any of their kids having freckles? Well, this person here is a heterozygote or a hybrid. So is this one, and so is this one, and so is this one. They all of them have one capital F and one lowercase f. All of them, all four of them. Which means they're all going to show freckles. So the odds of having a kid with freckles out of this crossing for these two individuals is 100%, or you could also just say it 4 out of 4. I don't care which way you answer. Typically, percentages are the easier way, but if you want to use fractions, that's fine. Nobody's going to argue with you. But let's change this a little bit. What would happen if we crossed two heterozygous parents? Two people who have one capital F and one lowercase f, and again, capital F and lowercase f. They're both heterozygous. So let's replay this. And what I'd like you to do is pause the video. I've set up the Punnett square for you. Again, one parent on top, one parent on the side, and see if you can work this out. What are the odds of the child having freckles now? Again, you can answer there in terms of percentages or in terms of fractions. I care not which. But just see if you can figure it out. Pause the video and then continue on. I'll wait a second or two before I continue. Okay, so let's run this through. If you did your cross properly, you bring capital F down, capital F over, capital F down, lowercase f over, capital F over, or again, capital F goes first, lowercase f down, lowercase f over. It doesn't really matter which one goes first in this case. But again, always put the capital letters first. But notice what's happening. Here we have a different outcome. The number of kids that potentially have freckles has changed. This child would have freckles, so would this child, and so would this child. Whereas this child would have no freckles. But notice the genotypes don't match that particular ratio. We have three distinct genotypes going on here. All three possibilities show up. There's a homozygous dominant. There are two, do this in a triangle here, heterozygous, just to really emphasize the difference here. And there's one that's homozygous recessive. So while 3 out of 4 of these would be showing freckles, the ratio for the genotype is different than the ratio for the phenotype. So let's clarify this. This here on the right is showing the genotype ratio. So this is, let's do this in black here. This is genotype ratio. I would probably label it if I were you. I forgot to label it on my slides. Sorry, my handwriting, I'm trying to write with a mouse. So this is the genotype ratio. The phenotype ratio, however, is different here. The phenotype ratio wouldn't match these numbers. Because again, the genotype you have 1/4, 25%, is homozygous dominant. 2/4 are heterozygous, and 1/4 is homozygous recessive. That's the genotype ratio. The phenotype ratio is which kids would show the trait we're looking at, in this case, freckles. And in that case, we have 3 out of 4 with freckles. And we also have 1 out of 4 without freckles. I'll just put a big capital F here just to clarify. So yeah, this person has freckles. Go to my pointer because you can't see my thing here. This person has freckles. This person has freckles. This person has freckles. That's their 3 out of 4 with freckles. And then 1 out of 4 has no freckles. You could say no freckles too. Notice that the ratios must always add up to either 1 or 100%. Again, this would be like 25%, 50%, 25%, and that's 100%. And then this is 75% and 25%, and that would be 100%. So the genotype ratio and the phenotype ratios don't always match. You shouldn't always expect them to. Sometimes they do, sometimes they don't. Just keep in mind that genotype is genetic. What letters are you talking about? Phenotype is physical. What's being expressed? Are you showing the trait? Are you not showing the trait? What's going on in the actual trait? But this ratio, this 1 to 2 to 1 genotype ratio, always occurs with a cross of two hybrids. And let me erase my right in here so this is easier for you to see. So when you have two heterozygous individuals, this 1 to 2 to 1 genotype ratio is to be expected. And again, it'll usually be it'll always also be a 3 to 1 phenotype ratio. 3 being the dominant trait, 3 being the dominant trait, and 1 out of 4 being the recessive trait. Now, this all came about thanks to a gentleman by the name of Mendel. And if you didn't study Mendel in middle school, let me give you a very, very brief breakdown. Mendel was actually a monk working in a monastery, I want to say in Austria, in the mid-early 1800s. He did a lot of work in the garden, and as he was studying his garden, he noticed that some traits in his pea plants showed up more often than others. And you think, well, what the heck does pea plants have to do with anything? Well, he was actually able to start predicting when a trait would show up. He actually had the time to sit there and work out the mathematics. And this is what he discovered. All these traits here on this left side turned out to be what he called dominant, more or less. These are the ones that showed up most often, whereas the traits on the right, they showed up on occasion, but not nearly as frequently. Thus, they were, as he determined, recessive. And keep in mind, he wasn't learning from anybody else. He was just doing this as just pure observational work. And he figured out for each situation how the mathematics behind this worked out. And it wasn't until much later in the 1900s when his work was actually rediscovered by scientists that Punnett came along and came up with this idea of showing it via Punnett Squares. So Mendel actually never did a Punnett Square. Punnett didn't show up till at least 60 years after Mendel's work was lost and then refound. But let's look at some of these that Mendel had originally talked about in his notes. Let's look at specifically seed shape, where round seed shape is dominant to wrinkled. And let's look at flower color, where purple flowers on pea plants is dominant to white flowers. And let's see if we can practice some more on Punnett Squares. Okay, so again, in pea plants, round seeds are dominant. We're going to use capital R. Wrinkled seeds are recessive. We're going to use lowercase r. Guys, again, it really doesn't matter what letter you use. Just don't get yourself confused on uppercase versus lowercase. You know, if you have letters that look very similar, uppercase versus lowercase, like P's and W's, just be very clear with yourself on how you write that stuff. So let's do this cross. We have one pea plant that is heterozygous, 4 round seeds, and one that is homozygous recessive. Again, make sure you start learning what these terms mean very quickly because they're going to be big deals. So this is our cross. I'll pause for a moment, try to do the Punnett Squares, and if you can, try to give me both the genotype ratio and the phenotype ratio for the outcomes. What is the genetic ratio for each of the offspring? What is the physical ratio for each of the offspring? And I'll pause for a second and I'll go on in a moment. So you may not have done your Punnett Square exactly like I did mine. You might have a capital R, lowercase r on the left side and then 2 lowercase r's on the top, and that would work out just the same. But again, one parent goes on top, one parent goes on the side. We do our bring down and cross over, so bring the capital R down, lowercase r's comes over, lowercase r comes down, capital R's come over, and this is our result. And again, if you changed where the parents were, you might get a slightly different outcome, but the actual odds should be the same. In this case, you can see here, 50% capital R, lowercase r are going to be round because they're heterozygous. 50% are going to be wrinkled. This, again, is our phenotype ratio. Label that. Our genotype ratio, however, let's confirm that. Is also going to be 50-50, but it's 50. Let's be absolutely perfectly clear. It's, let's do this in fractions for this time. 0 out of 4 are homozygous dominant. Nobody is 2 capital R's. 2 out of 4, that's a 2 out of 4, are heterozygous. And you should try to write the whole term every time. If I shorthand it, that's just because I'm writing with a mouse. You guys don't have that excuse. So here's my 2 heterozygous. And then another 2 out of 4 are my homozygous recessive. And again, you should write out the full term. I'm just shorthanding because you don't want to sit here waste more time watching me draw with the mouse. And so here's my homozygous recessives right here. So here the ratios, they look similar, but make sure when you're answering, especially genotype ratios, you give a percentage for everything, even if the answer comes out to be 0, 0% like up here. So just make sure you're absolutely clear with yourself. It makes things easier to prevent mistakes. Let's try another one. Again, sticking with round seeds versus wrinkled seeds, round being dominant. Let's cross these 2 parents, homozygous dominant and heterozygous. Let me go to my pointer again. So I'll pause, give it a moment, see if you can give me both the genotype and phenotype ratios for this outcome of this Punnett square cross. Okay, let's see what you got. Again, it might look a little bit different, but I put this parent on the top. You might have put them on the side, and that's fine. And then I put this parent over on the side. And then again, bring down, cross over, bring down, cross over, and we get this result. 2 of these parents are going to be homozygous dominant. So right off the bat, let's make that ratio up. So this is our, again, genotype. So homozygous dominant, let's say both capital letters, is 2 out of 4, or 50%. Heterozygous, again, write the full terms on your own when you do this. Don't shorthand like I'm doing. I'm just doing that because of the mouse. Heterozygous would also be 2 out of 4, and that leaves homozygous recessive as, let's put an equal sign here so I don't get confused, recessive as 0 out of 4, just to be absolutely clear. And then for the genotypes, well, here's again where the genotype does not match the phenotype, because if you look at the phenotypes, which ones are round versus wrinkled, 100% of them are going to be round, and 0 out of 4 are going to be wrinkled. So again, the genotype does not match the phenotype ratio. Again, phenotype is physical, genotype is genetic. So yeah, all of these, because they all have at least one dominant allele, will be round. It's just that these ones are homozygous dominant and these ones are heterozygous. So make sure you're clear on the difference between genotype and phenotype ratios. Let's try another one, and we're going to switch traits now to a slightly more prettier and yet somewhat more difficult one, flower color. So let's suppose we have in pea plants purple flowers and white flowers, which is reality for how this works. Purple flowers are dominant to white. And let's cross these 2 parents. Notice I'm not giving you the letters this time other than to use the letter P. So I'm going to pause it, see if you can work out the Punnett square and get both the genotype and phenotype results from that. Ratios, I should say. Okay, so let's see if your answers match my own. I put the homozygous recessive parent on top. Homozygous means the same. Recessive means both lowercase p. So lowercase p, lowercase p. Heterozygous parent means one uppercase, one lowercase, and I put that one on the side. Uppercase, lowercase. And then again, you do your crossing. Just make sure you keep it clear what's uppercase and what's lowercase. So bring the uppercase over. I did that one first because it's the capital letter. Lowercase down, and then lowercase down, lowercase over, lowercase down, lowercase over. So here, the odds of getting the odds of getting a heterozygous, and let's do my pen again for my genotype. So I'm just gonna put gene. Genotype. So the odds of getting, well, let's do homozygous dominant first. Let's be absolutely clear. Should know better by now. Homozygous dominant, the odds are 0. 0%. For a heterozygous, because none of them are both capital P's. For a heterozygous, the odds are here and here, so that's 50%. Notice I'm using percentages this time, not fractions. Again, I don't care which one you use, just make sure you're right. So 50%. And then homozygous recessive, again, when you guys do this, write out the full terms, don't shorthand. Homozygous recessive is here and here. So again, that's 50%. And again, this should always add up to 100%. So that's my genotype ratio. As for my phenotype ratio, I would have 50%, these top two, here and here, being purple flowered, and these bottom two being white flowered. And again, has to add up to 100%. And again, this would be my phenotype ratio. Sorry, it's not labeled. Phenotype. Now, in reality, it is very simplistic to think of genetics in this terms. This is a simple model. This is like me first describing a car to you by first showing you something like a Hot Wheels. It looks like a car. It might have parts that act like a car, but it's not really a car. It's way oversimplified. And this is true for how Punnett squares work as well. They don't always work like this, and in fact, most times Punnett squares don't work for many traits because most things are not dominant or recessive. And then it gets really tricky when you try and consider mixing two traits. So suppose you have one parent, let me switch. Suppose you have one parent who has round seeds, who's homozygous dominant for round seeds, and who's also homozygous dominant for purple flowers. This is just one individual, but we're now talking two traits. Whereas they're crossed with another parent that has wrinkled seeds, homozygous recessive, and has white flowers, again, homozygous recessive. When you start adding multiple traits together, the nature of the Punnett squares gets more complex much, much faster than you expect. Because to do this as a Punnett square problem means you'd have to set up a square system of not just 4 squares, but of 16 squares in reality. And let me show you what I mean. This is what we call a dihybrid cross. And I'll put that label here. This is what we call a dihybrid cross. Di because there's 2 of traits being discussed. Okay. So this one is actually has a nice easy shortcut that you won't always see. You could recognize that, well, this individual can only pass along a capital R, and this individual could also only pass along a capital P. There's no other option. There's no other possible combination. Whereas this individual could only pass along a lowercase r, and this individual could only pass along a lowercase p. Again, there's no other option because it's homozygous as well as this is being homozygous. Once that's clear, you don't actually have to do a Punnett square. Even though it's going to get more complicated, you wouldn't have to do one in this case because you know that the results are always going to end up being the same. You're going to have a heterozygote for both traits, capital R, lowercase r, capital P, lowercase p. And 100% will have round seeds with purple flowers, and it'll be a heterozygous all the way across. But again, that shortcut only works in this situation. For a real situation, this is what we call a true dihybrid cross, where you have a heterozygote for both traits crossed with another heterozygote for both traits. And again, the square system for this is going to come out to be 16 squares, and it's going to be on the next slide. So if you want to try it on your own, you can, but otherwise, make sure you pay attention. Okay, so let's try this problem, but again, it's going to become a much more complex Punnett square, this dihybrid cross. And again, to do this properly, you would need actually 16 squares to solve for everything. And again, you're going to put one parent along the top, one parent along the side, and let me show you how that should work out in this situation. Now, how did I get these for each side? This is where we want to refer back to a system in mathematics, which if you're learning it, called FOIL. That works the best, I find. Whereas, where's my mouse cursor? There we are. This first one right here, capital R, capital P, first, because these two can be paired together. Then capital R, lowercase p, outer, and inner, this one, and last. So you could have capital R with capital P, capital R with lowercase r with capital P, and lowercase r with lowercase p. And then I've just repeated that same thing along the side here because this parent on the right is the exact same as the parent on the left. So using that FOIL method, first, outer, inner, last, gives you all possible gamete connections. This is part of something called the law of segregation, which and independent assortment, excuse me, which we'll get to later on. But once you have that, then you would have to do your whole bring down and crossover routine again for a Punnett square. So you would have to, bring down, I'm not going to do this all for you, but, you bring down a capital R, then bring over a capital R, then bring down the capital P, then bring over the capital P. You keep the letters next to each other. They're like the same letters. Keep all the R's next to each other. Keep all the P's next to each other. And remember, capital letters go first. So like for this one down here, you would bring down a capital R, and then there'd be a lowercase r, then you'd bring down a capital P, and there'd be a lowercase p. And you'd have to fill in for all of these Punnett squares. But even after you do that, you do find certain patterns. Look at what I mean. Each of these, again, the R's are kept together, the P's are kept together, capital letters always go first, so like here. You do see certain trends appearing. For instance, this is homozygous dominant across the board, and this is the only one that's like that. Down here at the far opposite corner, this is homozygous recessive across the board. This is the only one like that. Then these two here and here, those two are the only two that have that specific makeup, where it's homozygous dominant for the first trait of round seeds and homozygous recessive for the second trait. And then again, homozygous recessive and then homozygous dominant. So this diagonal here is unique. But there are patterns. I've color-coded that so you can see. So this is homozygous dominant, then heterozygous, and it's the only two that are like that. And you could break this down in terms of, I'm looking at the genotype ratios here. You could break this down in terms of phenotype ratios. So how many have round seeds with purple flowers? How many have round seeds with white flowers? How many have wrinkled seeds with purple flowers? How many have wrinkled seeds with white flowers? And break it down that way too. But there are still patterns to be found here. So you could be asked to do, again, genotype and phenotype ratios for this entire thing. And there'd be a lot of possibilities in some cases, especially for the genotypes.